Hello and welcome, dear students. How are you all? I welcome you all to the class of biology. So, dear students, today we are going to continue with where we left off in the last class. All right. So, in the last class we had studied about cell. Okay. So, how many people of you remember what I had given you for the homework? Students, please report your attendance. Itanagar, Tamenglong, Shillong. Good afternoon, Shillong students. How are you? Yeah, so we are going to start the chapter now from where we left off in the last class. Okay, Itanagar 1 students, all right. Tamenglong, all right. Good afternoon. How are you? Shillong students, please report attendance. I believe your camera is not working right now. Shillong. Okay, yeah. So please, in the meanwhile, report your attendance. Is your camera not working, Shillong students? Yeah, okay. So, alright. Meanwhile, okay, yeah, now I can see you. Yeah. So we were doing the cell theory, the cell chapter we were doing after completing the tissues, the anatomy of plants. We are now completing the chapter called the cell. Yeah. Centers, please report your attendance, everyone. Only Shillong students, I believe, are remaining, reporting their attendance. Right? Okay, yeah, please settle down quickly. Alright, so let's start now. I had given you some homework. I had asked you to find out about the largest cell and the smallest cell. And I asked you to write about the largest plant cell also. So have you done it? Have you done it? Okay. So these are the some of the largest cells that I wanted to tell you about, right? So I have these things for you. I believe you will find these interesting. They should interest you in any case. Okay, so yeah, what we have here, this is the largest cell of the ostrich. Okay, see on your screens. Yeah, so we have the largest cell of the ostrich right here. Okay, so just a second. Yeah. For comparison, you can see here, this is the ostrich egg. This is the ostrich egg, largest animal cell, right? This is the egg of a hen's egg. You can see the difference in the size. Okay? You can see the difference in the size here, right? So hen's egg is really very small and ostrich egg is really huge, right? So I guess for all the students of the Shillong that are sitting there, they will enjoy an omelette made from ostrich egg, and it should be enough for every one of you, right? There is a large amount of yolk in that, so I believe the omelette that you can get from it should be enough for every one of the students that are sitting in the class, right? It's really huge. Okay, so about 10 people, around 10 people can eat from the egg. That serves a breakfast for about 10 people. 10 to 12 people I have heard. Alright. So what is inside the egg? Inside the egg is the albumin, the egg white, and the yolk. What is the yolk? Yolk represents the swollen ovum. Yolk represents the swollen ovum. Okay. Yeah, I'll send this picture in your group later. Okay. I'll send this picture in your group. Just wait a bit. Okay. So, because I'll have to send it to everyone. So, wait a bit. I'll send it in your groups later. Alright. And the smallest cell, mycoplasma, it is also shown here. Okay. It's under the electron microscope or the very high power magnification light microscope. Alright. Now, let's come on to largest plant cell. Largest plant cell. So this is the largest plant cell, which is caulerpa. Largest plant cell, single plant cell, single. This is caulerpa. Caulerpa. Okay. There is another one which is called acetabularia. I will send that picture in your group also. Okay. But this is the largest plant cell, which is caulerpa. Okay. Now for comparison, I'll give you one more picture. For comparison, I will give one more picture to you. Okay. Yeah. So the bacterial mycoplasma, it is about 0.0001 millimeter. The mycoplasma that you just saw. Red blood cell is 0.009 millimeter. Liver cell 0.02, it's larger. Human egg, yes, human egg is the largest cell in human's body. Okay. In case of females, they have the largest cells found in all the humans, and that is the human egg. It is 0.1 millimeter. Hummingbird egg, for comparison, is 13 millimeter. Hen's egg is about 6 centimeters or 60 millimeter. And ostrich egg, the biggest cell is about 170 millimeters. Okay, so it's really huge. Alright, so we have done this much. Alright, so now who will give me the revision for last time? The last time we had just started cell, that cell is the unit of life and this and that. So let us start that with a quick PowerPoint PPT I made for you. I hope you will find this interesting. Okay, just a second. So I'm going to play this, and right, hope you find this interesting. Alright, I hope you don't get bored enough now. Alright, so when we talk about the cell, in this figure there are various types of cell. The red colored picture on the left hand side, okay, that is a red blood cell. And beyond that, above that, left hand top corner, it is the animal cell. Okay. There is another cell painted in blue for this comparison. It's a very small cell. Okay, it's a mycoplasma. Then there is a plant cell. Then there is a bacterial cell. And there is a normal eukaryotic cell with all the cellular components. Okay. This is just a picture, no need to write anything here. So what did we discuss last time? We discussed about cell theory given by Schleiden and Schwann. Okay. Schleiden and Schwann stated that all organisms are made of cells. Cell is the basic unit of life in all living things, and all organisms, their cells arise from pre-existing cells, right? Come from pre-existing cells. Cell theory consists of just 2 points. Fact number 1 and 2, they can be said as one fact also, or they can be put as 2 separate facts. So this is about the cell theory. Okay. So I already taught you this. I will send this PowerPoint presentation in your group. Don't worry about that. Okay. I will send this PPT to everyone. So this shows that all living things share a similar unit of life. Okay. All living things share a similar structure. They have a similar building block that is common to everyone. Okay. So all eukaryotes have a common unit structure. They might look different from the outside, but an animal cell of one organism is quite similar to the animal cell of other organisms, even like amoeba. Right? Basic cell organelles and structural components are all the same. Only difference is in the DNA. Now, there are 2 basic types of cells. One is the prokaryotic cell and the other is the eukaryotic cell. Right? So whether it is a prokaryotic cell or it is a eukaryotic cell. Now, if you see in this picture, which cell is larger in size? Which cell is larger in size, students? Which cell is larger according to you? The eukaryotic cell, right? So the eukaryotic cell is larger, and it is much larger than what is shown in this picture. You can say prokaryotic cell is about the size of mitochondria of eukaryotes. This is the difference in size. Eukaryotic cell is very much larger. Prokaryotic cell is very much smaller. Now, we have to, in this chapter, what we are going to do today, we are going to study about organelles today, that what are the basic organelles that make up all the living systems, right? So these are the organelles that we are going to study today. Alright, now this is just a short summary. Take a look at it. Prokaryotic cells, they have no membrane covered nucleus, no membrane covered organelles, have circular DNA. Mostly they are bacteria, alright. And there might be some mycoplasma also, there might be rickettsia, chlamydia. All these are prokaryotic cells, even archaebacteria, right? So basically these are bacteria, okay. So this is, on the right hand side there is a very dark colored image, that is a electron microscope view, and that electron microscope view in this figure, it has been magnified and improved in a form of a diagram. So in the diagram you can see the picture more clearly, okay. Alright, so I will send this, no worries. Eukaryotic cell, they have a nucleus, they have a membrane covered organelles, they have linear DNA, okay, no, not a circular DNA, and all other cells are made of eukaryotic cells. Okay, now continuing with organelles. So what are organelles? First cell organelle that we have to study about today is the cell membrane. So cell membrane we had not started with in the last class, okay. So what is cell membrane? Cell membrane is a gateway, it's a pathway, it's the outer layer of the cell. It allows the nutrients into the cell and the wastes outside of the cell, right? It's a gateway, right? Just like there is a door in your classroom, okay? So it's a gateway for the things to enter. It's an outer barrier, okay? So it's a gate into the city or into the eukaryotic cell or the plasma membrane. Then what's found inside? Inside we see the cytoplasm. It's a jelly-like fluid and it's contained in the cell which holds the organelles. So it's a normal thing, nothing special about it. Everybody knows about it. Now let's come to the nucleus. Nucleus is the control center of the cell. It's the most important organelle of the cell. Okay, it contains the cell's DNA. Inside the nucleus there is another structure called the nucleolus. Okay, so this is a very important point, and you can see in this picture there are some dots. There are some pores inside the nucleus. In this figure, in the nuclear membrane you can see it is double layered, right? You can see that the nuclear membrane is double layered. So in this double layered membrane you can see there are many pores inside that nuclear membrane, and these pores are called the entry points and exit points, okay. So you can compare it to the very important thing in your city also, like there is a mayor in your city. So this is just like the mayor's office. Everything that has to happen inside that city or the cell, it is going to happen through the mayor's office, right? Next comes to the mitochondria. In the mitochondria you can see there is a extensive folding of the inner membrane. There is outer membrane also. So just like nucleus, this is a double membraned organelle. It's a double membrane structure. It's a power center of the cell. It provides the energy to the cell for everything. Okay, so it's a power control center of the cell. Without the mitochondria, you cannot have anything. Mitochondria is a basic control point because it controls the energy requirement of the whole cell. Okay, you can see, compare it to the electric company of the city or the cell, right? Next we talk about ribosomes. Ribosomes are the protein factories of the cell. They are made of proteins and ribosomal RNA. We'll discuss this. This is a protein factory of the cell. Next comes the endoplasmic reticulum. Endoplasmic reticulum is of two types: rough endoplasmic reticulum, smooth endoplasmic reticulum. So rough is having a ribosome, smooth is does not have any ribosomes. It is a transport system of the cell, and smooth ER, there are no ribosome, just like that. Okay. So anything that has to go out of the cell, the instructions will be passed from nucleus to the endoplasmic reticulum. So it will be making things, it will be manufacturing things, and it will pass on these things to the Golgi body. This is the Golgi complex or the Golgi body. Look, whatever thing is coming out of the cell, it has to be packed first. Suppose you receive an iPhone from your friend or your family, somebody gives you an iPhone on your birthday, so it will be a very good day for everyone, right? So suppose somebody does that and they give you an iPhone without the package. They don't give you the box of the iPhone. They say they give you a new iPhone. This is the iPhone, this is the charger, this is the power adapter cable, this is the headphones, the wireless headphones, and they don't give you any box. So it will be very irritating for you, right? So whatever things we give to anything, we must first tell what is the thing that we are giving. Okay. Suppose somebody gives you a different phone, but you don't know which company that belongs to, right? There is no information because you don't have any package. So that might not be accepted well by everybody, right? So Golgi complex is a similar thing. It is a packaging and processing industry, packaging and processing house of the cell. Without this, there is no information given about molecules will be going where to which site. So it packs and processes and it ships the things in proper packets. It's going to package it in a membrane, in a bag, okay, and then only the things are going to reach their target destinations properly. This is the function of Golgi complex. I'll explain this. Okay, all right. Lysosome. Lysosome, basically they are the recycling centers. They digest food particles, cell parts. They can even digest old mitochondria, old organelles, non-functional organelles. Okay, so these are basically recycling centers. They contain a lot of enzymes. They can digest everything, and they can also be called suicide bags. If they explode inside the cell, they're going to destroy the whole cell. Okay. Next is the cell vacuole. So cell vacuole is the largest organelle in the cell. You can see 90% of the volume, nearly, it is occupied with a vacuole in case of plant cell. Okay, so it's occupying a very large space. Okay, and it stores water, food waste also. So basically, this is a garbage collection system also, and this is a water reserve also. Okay. So if the vacuoles are filled with water, the cells are turgid. If the vacuoles are empty, cell volume will decrease very much and the plant cells will drool down. In this figure you can see, right, the plant is standing erect, right, standing erect when the vacuoles are full of water. If the vacuole is shrunken up, cell volume will decrease. So the whole plant is wilted. This condition is called wilting, W-I-L-T-I-N-G. Next comes the cell wall. Now what is a cell wall? Cell wall is found only in plant cells or in fungal cells or in bacterial cells, right? Sometimes in protista also. So it protects and supports the cell, okay? And can you tell me any product of cell wall that everybody is using right now? Can you tell me any cell wall product that you are using right now? Anybody? Without that, you won't be able to sit in that class. Give me a name of cell wall product that everybody is using in your class right now. Wood, yes, but there is one more important thing than wood. Without that, you cannot come to the class. Wood is the correct answer, of course, but there is one more thing. Without that, you cannot come to the class. Clothes. Your clothes are made of cotton. Cotton is seed, coat, testa, cell wall. Okay, you're basically wearing a cell wall of cotton, right? So everybody remembers the importance of clothes, right? So without that, you cannot come to the class, right? Not the copy, not the wood. There's basically something more important than all these things, the clothes. All right. So cotton is 99.9% pure cellulose. It's basically made of just of the cell wall only. That's the only thing. All right. Chloroplasts. Chloroplasts are found in plant cells. They contain chlorophyll, makes the plants green. This is the place where photosynthesis takes place, right? So I'll explain the structure of chloroplast in full detail. Differences between plant and animal cell. things there are found in both plant and animal cells. These are mentioned on left side. Things mentioned on the right side are found only in plant cells. Okay. All right. You will be asked in exam also what the differences between plant and animal cells. I hope you have done it. I gave you this as homework. Okay. So this was the first thing that I wanted to show you. Okay. All right. I have one more slide. That was a shorter version. This is a longer version. I hope you can see this now. So, discovery of cells was by Robert Hooke, and in this slide I have even included the cork slice section. This is a cork slice, the thing that Robert Hooke had drawn in his diary. This is that structure. This is a thin section of cork. Okay, so these are the empty boxes, or from which cell term was coined. Now, cells have a very large surface area to volume ratio if it is a small cell. But if it is a large cell, okay, then the surface to volume area, surface area by volume ratio is going to decrease. Okay, so here we have a cell. Okay, on the left hand side, it's a larger cell, and then on the right hand side, I am going to make many cells, 8 cells from this smaller cell. So you can see on the right hand side, the smaller cells have a larger surface area to volume ratio. Larger cells, they will have a total overall larger surface area, but compared to the volume, it will be very less. 3:1, 6:1. Smaller cells are more efficient. Alright, so this is what everybody knows. Okay, prokaryotic, eukaryotic, I have already described. This is second slide I prepared. Okay, so I will give you this also. In this, I have included more diagrams. So I hope some people will find my other slide more interesting, or some people will find this more interesting. This contains more details. Okay, and this is what we are going to study today. Okay, I will give you the notes regarding this only. So let's start, and we will do this side by side. I'll show you, keep you showing these diagrams as we move on. So let's start today's class. We had done up till prokaryotic cell. Okay, prokaryotic cell we have already discussed, but I'll give you a quick recap. Prokaryotic cell. Now what's a prokaryotic cell? It's a primitive nucleus containing cell. Or we can say that a prokaryotic cell is the first cell. Cells are of, look, I have already told you the cells are of two types: prokaryotic and eukaryotic. So what we are going to cover today is starting from prokaryotic cell to the eukaryotic cell, and then plasma membrane, chloroplast, mitochondria. We have to cover a lot. So prokaryotic cells do not have a well-defined nucleus. Do not have a well-defined nucleus. They do not have any membrane-bound cell organelle. They do not have any membrane-bound cell organelle. Right? Prokaryotic cells are smaller than eukaryotic cells. Right? You already know that. They are smaller than eukaryotic cells. they are smaller than eukaryotic cells. Okay. All right. So I'll just keep scrolling and writing. The prokaryotic cells, they have some things like ribosome, but ribosome is a cell organelle without having any plasma membrane. Okay. They have flagella also, and flagella are mostly, they are covered with plasma membrane. Flagella, they are covered with plasma membrane. But please remember, when somebody asks you, do prokaryotes contain any membrane bound cell organelle? Then always you will say that no, prokaryotes do not have any membrane bound cell organelle. Alright, this is the fact. Okay, they contain 70S ribosomes, 70S ribosomes. Cell wall also they have. Most bacteria, most prokaryotes have a cell wall. Cell wall they have, but they are mostly unicellular, no colonization, no multicellular structure is present. Colonies might be present sometimes, but no multicellular structure. They have a cell wall. Other than that, they have nucleoid or naked genetic material. Okay, nucleoid or naked genetic material. Anything else you want to add here? Nobody wants to add something here? What do they contain else? Mesosome, very important. Other than that, what do they have? Flagellum might be present, might not be present. Flagellum might be present. It's quite common, so that is why I'm writing it. And what else? They contain some inclusions, some granules. Sometimes they contain phosphate granules. Phosphate granules and synophycean granules. Synophycean granules. And what else? Gas vacuoles also. But these gas vacuoles, please remember, the phosphate granules, synophycean granules, none of these granules, they have any plasma membrane. They do not have any plasma membrane. Okay? So when I say that prokaryotes do not have any plasma membrane bound cell organelle, it is the absolute truth. Right? Etc. Alright? So first of all, let's discuss the cell wall. The cell wall of prokaryotes. So what is the cell wall of prokaryotes made of? What is the composition of cell wall of prokaryotes? Look, when I taught you kingdom Monera, the prokaryotes, so I had given you this information regarding what is the composition of cell wall of the prokaryotes. So please, I hope you remember. Please tell me quickly, what is the composition of cell wall of the prokaryotes? Nobody. I have given you this information when I taught you kingdom Monera in the biological classification chapter. Okay, if you don't remember. The cell wall of prokaryotes is made up of the cell wall of prokaryotes is made up of N-acetyl glucosamine, N-acetyl glucosamine, NAG plus N-acetyl muramic acid. NAM. The cell wall of prokaryotes is made up of NAG plus NAM. It's a cross-linked polymer. And do you also remember, together it is called peptidoglycan. Together it is called peptidoglycan. Now, do you also remember what is the poison, what is a kryptonite to this superstructure? What thing can break this cell wall? You want me to write in bigger handwriting? Okay, I believe, I can do that, no problem. I believe the page was a bit, okay, I'll do that, no problem. N-acetyl glucosamine plus N-acetyl muramic acid. Okay, I hope it is visible now. Right, Shillong students? Now I am asking you, what can break the cell wall? What is the poison to the cell wall? Lysozyme. Lysozyme, it is found in human tears, human saliva, human secretions, right? So this enzyme can break the bacterial cell wall. This is the reason why animals lick their wounds, because their saliva has antibacterial property. So if they keep licking, the bacteria will not be able to grow. Right? This is the reason. Okay. Now after cell wall, bacteria, almost all bacteria have cell wall. Archaebacteria have a different cell wall composition. Mycoplasma do not have any cell wall. Okay. So all this information I have already given you. So there is no need to repeat that. Right. Now let's come on to, after a cell wall, let's come on to the nucleoid. Nucleoid or the genophore or the incipient nucleus or the primitive nucleus. All right. These are the alternative names. I hope everybody can write all this. Yeah, so I'm writing capitals also, so that some things are clearly visible. Okay. So this is the genetic material of bacteria. The genetic material of bacteria or prokaryotes is loosely packed with the help of non-histone proteins. Okay, non-histone proteins make up the nucleoid or the primitive nucleus. Okay. It contains DNA. It contains circular DNA, and I'm writing a term for it, DSCCC, double-stranded circular covalently closed DNA. If anybody is left, let me know. If there is something that you have missed, please let me know. Okay. I hope everybody has written it. So the DNA, it is not linear. In eukaryotes, the DNA is linear, but in prokaryotes, it is closed in the form of this line is no longer a line, it has become a circle. So that is what I am calling a circular DNA. Both the ends have joined together, and that is by a loose covalent bond. Covalent bond. It's not very loose. Double-stranded and circular and covalently closed DNA. This is the type of DNA that is found in bacteria. After that, please put the heading mesosome. Mesosome. So what is a mesosome? What is a mesosome? If I draw a bacterial cell, like this, then what will happen is, this is the primitive genetic material. This is the primitive genetic material. Like this. And this is the mesosome. So this structure is the mesosome. These are 70S ribosomes. 70S ribosomes. This is the nucleoid. This is the cell wall. This is the plasma membrane. Or the bacterial cell membrane, plasma membrane or bacterial cell membrane. Okay. This is the basic structure of a prokaryotic cell. Okay. Please finish it quickly. Okay. All right. So let me continue about mesosome. What's a mesosome? Mesosome is an infolding of the plasma membrane to increase surface area, increase the surface area you want me to slide up. Okay, please finish it quickly. Some people I believe have not been able to write it, so I'm sliding it up. Do it quickly, please. Okay. Itanagar has not done. Itanagar, have you finished? Others have done. Again, what have you not? What is remaining, Itanagar? I believe I have done this much. First one. You want me to open to this slide? I had already, yeah, okay. If you have not, alright, please finish it. Okay, of this heading, sir, heading of this, okay. Now it is done, Itanaga. I have already, yeah, okay. Look, these things I have already discussed with you. These titles I have already written in Kingdom Monera. So if you are missing this, you must go to my previous lectures of Kingdom Monera. There I have given this. Okay, so mesosome is an unfolding of the plasma membrane to increase the surface area. the enzymes associated with respiration, the enzymes associated with respiration are found on mesosome. Associated with respiration are found on mesosome. Okay, the enzymes associated with respiration are found on mesosome. So this means that it could be a precursor of mitochondria, or it's equivalent to mitochondria. Therefore, it is considered to be equivalent of mitochondria in prokaryotes. Therefore, it is considered as equivalent of mitochondria it is considered as equivalent of mitochondria in prokaryotes. All right. It's considered as equivalent of mitochondria in prokaryotes. It is closely associated with nucleoid. It is closely associated with nucleoid so believed to help in binary fission. It is believed to be helpful in binary fission. It is believed to be helpful in binary fission. Okay. So mitochondria equivalent is mesosome. It contains enzymes for respiration. It is believed to help in binary fission. These are the functions of mesosome. So it's very important that you remember about mesosome. Okay, it's an enfolding. It contains the respiration enzymes. Next, move on to, we have done this much. So now we will do what's remaining. Cell wall is done. Okay. So cell wall is done, nucleoid is done, mesosome is done. Now we have to do flagellum and 70S ribosomes. Ribosomes I will cover later. So ribosomes will be covered in eukaryotes also, like cell organelles. So ribosomes are nothing special. I'll write a short note on that. We have to do flagellum and granules, and then we will move on to the eukaryotic cell. Flagellum of bacteria. Flagellum. So you can see here, a flagellum is made of flagellin protein. Flagellin protein it may or may not be present in bacteria. It may or may not be present in bacteria. It may or may not be present in bacteria. If it is present, then there is one. Then we have many different types of flagella in bacteria. But if it is not present, it is called atrichous. If it is not present, it is called atrichous. If it is present, monotrichous, single flagellum, two flagella on opposite sides, amphitrichous, flagella on all sides, the cephalotrichous, the peritrichous, and then a bunch of flagella on one side, lophotrichous, cephalotrichous. Alright, so all these things we have covered in kingdom Monera. So I am not going to discuss this again. Flagellum helps in movement of bacteria, helps in locomotion. So it is the locomotory organelle. Locomotory organelle. Locomotory organelle. Okay, so it is going to help in locomotion. It's a locomotory organelle. All right, so yeah, one more thing. After flagella, we will write a short note: 70S ribosomes. 70S ribosomes. Okay. So what are the 70S ribosomes like? These are non-membrane bound organelles. Flagella is covered by cell membrane, but it is not covered by plasma membrane. Non-membrane bound structures. Non-membrane bound structures. They are composed of two subunits. They are composed of two subunits. One is the larger subunit. Called the 50S. Smaller subunit is the 30S subunit. Now there is no arithmetic addition rule in ribosome. Ribosome is made of 50S plus 30S, but if you add them together you will get 80. So there is no arithmetic addition rule in ribosomes. If you add this to this, you will not get 80S ribosome. 80S ribosome is found in eukaryotes, and 80S is composed of different subunit. It is composed of 60S and 40S subunit. There is no arithmetic addition rule. Please do not add them together and don't get confused. Alright. 70S ribosome is made of two subunits. One is called 50S, other is called 30S. Just write this right for now. There is no, this is something to be learned. Please do not try to apply any concept here. Okay. This is about ribosome. Then there are some granules. Granules we have to talk about. So there are some phosphate granules. Phosphate granules. These are phosphate reserves found in the cell. Nothing special about it. Phosphate reserves found in the cell. Synophycean granules. Synophycean granules. Synophycean granules. So what are the synophycean granules? These are found in Synophyceae. These are found in Synophyceae or blue-green algae. Blue-green algae. Okay, these are pigment granules. These are pigment granules. They are pigment granules. These are not also covered by plasma membrane. No plasma membrane around these structures. Last is gas vacuole. These are meant for buoyancy. These are meant for buoyancy. Okay, buoyancy means ability to float in the water. They are meant for buoyancy. Okay, they are meant for buoyancy. They help the prokaryotes to stay on the surface of water. To stay on water surface. Stay on water surface. Right? So this is all about prokaryotes. Now please put the heading eukaryotic cells. Now we are going to start eukaryotic cells. So I hope you have done it. Is there anything remaining in this? And if you want these notes, I can also give you these notes. Okay, no problem in giving you these notes. You can take these notes as well. Okay, I hope that helps everybody, right? So everybody is writing these notes. Okay. Yeah, you want a break? Okay, no problem. All right. But my class is about to get over in about 1 hour. All right. So let's take a break. 5 minutes. Try to be quick. Okay, so we are back now. Right, so had your break? Alright. So now let's put the heading eukaryotic cells. I think I'll just do it on the next page. Eukaryotic cells. Okay. So when we talk about the eukaryotic cells, the eukaryotic cells, they have a lot of organelles, right? So this is a typical eukaryotic cell that you can see here in this figure, okay? And it has a nucleus in the center, and nucleus has a endoplasmic reticulum closely associated with it. In this figure you can see here, right? And inside the nucleus is another structure called the nucleolus. The nucleolus and the nucleus, they are within a single system of double membranes, and in the double membrane you can see some nuclear pores over here. See the labeling please. And besides that you can see some centrosomes, centrioles, microtubules, the cytoplasm, the ribosomes, and moving on further we have smooth endoplasmic reticulum, the free ribosomes, the mitochondria, the Golgi apparatus, the lysosome. All these structures are present. Okay. So let's start with the first structure. It is the plasma membrane. The plasma membrane. So can anybody tell me what is the difference between plasma membrane and cell membrane? Can anybody tell me what is the difference between plasma membrane and cell membrane? Anybody? Yeah, you want to take a guess? Take a guess. If you're wrong, I'll correct you. What's the difference between plasma membrane and cell membrane? Everybody must have heard of cell membrane, right? But now I'm asking you about plasma membrane. So what's the difference between plasma membrane? There is no difference. There is no difference between plasma membrane and cell membrane. Whenever the plasma membrane is present around the cell, it is called cell membrane. But if it is present around any cell organelle, it will be called that organelle's membrane, but it will still have a structure that is similar. All plasma membranes have similar structure. Mitochondria membrane, chloroplast membrane, lysosome membrane, nucleus membrane, cell membrane, all have equivalent structure, and that structure is called the plasma membrane. Now cell membrane or a plasma membrane contains cell contents. It is composed of a double layer of phospholipids and proteins. Okay, you can even say lipids also. It's that's perfectly fine. So you can say it's a double structure of phospholipids and proteins, or lipids and proteins. It's a bilayer. Now what are the phospholipids, or what are lipids? The phospholipids have a hydrophilic head and a hydrophobic tail. Water loving head and water hating or water repelling tail. Now the structure of this is, there is a phosphoric acid molecule here. It is hydrophilic part, and it is associated with glycerol, and glycerol is linked to two fatty acids. A single glycerol molecule, it is linked to one phosphoric acid molecule and two fatty acid molecules. This is called a phospholipid molecule. This phospholipid molecule it has a structure like this, that is shown here. It interacts with water, and this is what is going to build the structure of phospholipid plasma membrane. So now please put the heading plasma membrane. Okay, so the various organelles, of eukaryotic cells are as follows. Eukaryotic cells are as follows. Number one, plasma membrane. Plasma membrane. So what's the plasma membrane? It is the outer covering of all cells Plasma membrane is composed of two layers or bilayer of lipids and proteins. Plasma membrane is composed of bilayer of phospholipids and proteins or lipids and proteins. Okay. Now the structure of plasma membrane was explained by Singer and Nicholson using fluid mosaic model. The structure of plasma membrane was best explained by Singer and Nicholson using fluid mosaic model. Fluid mosaic model. Using fluid mosaic model. Right? So what is this fluid mosaic model we are going to study now? Look, the fluid mosaic model is here. This is the fluid mosaic model. Okay? We are going to draw this. So let's draw the structure of fluid mosaic model. We are going to have a bilayer of lipids, phospholipids and proteins. And inside this, there are some proteins like this and that here. Okay. Proteins are here. Like this. Okay. Like this. Right here. And inside this, there are some proteins here also. More than that, we are going to discuss some structures. Basically, we have to draw a zigzag tail like this. Okay. And there are some proteins here also on the outside surface, on the inside surface. Okay. Like this. So, now let's write about it. Integral protein. Integral proteins. Bilayer of phospholipids. Phospholipids and proteins. Okay. So the hydrophilic head faces outwards, the hydrophobic tail faces inwards. And all the structure, it is quite fluid. It can easily change its shape, okay, because it's a sea of lipids, and these lipids are very loosely arranged in respect to each other, so it's also quite fluid, and it is selectively permeable. Please write these things now. The plasma membrane is composed of large amount of phospholipids arranged in two layers. Phospholipids arranged in two layers. Okay. Proteins are present. Proteins are present in the lipid bilayer like icebergs floating in sea. Icebergs floating in sea. Okay. Now, in this structure, there are a few more things present here. These are glycosylation residues or carbohydrate residues. Okay. This is a carbohydrate residue, and this, when this violet colored thing is attached to lipid, it is called glycolipid. If violet colored structure is attached to protein, it is called glycoprotein. I'll explain what it is. Glycoprotein. Glycoprotein. And the other thing is glycolipid. There is nothing special, you just have to draw a fiber-like structure. Okay. So the plasma membrane has two layers, And proteins and lipids both are associated with carbohydrate structures. These are called glycolipids and glycoproteins. These function as receptors. Receptors are the basic functions performed by the receptors. Basic functions, they are performed by special glycosylate residues or carbohydrate associated with lipids and proteins. These are used for recognition. Just like you need an ID card for entering into the school, any protein, any receptor, any molecule, it also needs an ID card. Or cells need an ID card to understand each other. Look, all cells are blind. They do not have eyes. So how does one cell recognize another cell? It is done with the help of receptors. And receptors are formed by proteins associated with the carbohydrates called glycolipids. And this is how they recognize each other. Okay. So you can see this figure here. You can see there are many proteins here. Okay. And if carbohydrate residue attached to protein, it is called glycoprotein or glycolipid. All right. Clear? So the plasma membrane is quite fluid in nature. Therefore, it can easily change its shape. The plasma membrane is fluid in nature. And can easily change shape. Proteins can also move from one position to the other because they are floating, right? So they can also move from one position to the other position. Proteins can also move anywhere within the lipid bilayer. Proteins can also move to any place within lipid bilayer. Okay. All right. So this is all about plasma membrane. Okay. I hope you have done it. All right. So let's now put the heading girl apparatus or girl. Okay. So now let's put the heading girl. Girl. What is girl? What is girl? I think I gave you a small hint last time we started this chapter. Does anybody remember? What is girl? No, I'm not talking about females, right? I'm not talking about the females. Girl or endomembrane system. Endomembrane system. Girl refers to Golgi body, endoplasmic reticulum, and L stands for lysosome. L stands for lysosome. This is the expansion of the term girl. Now we are going to study this one by one. First of all, we have to study the endoplasmic reticulum. endoplasmic reticulum. Right? So what does endoplasmic reticulum refer to? I hope you have done it, right? So I'm shifting the slide down. I actually need to make a diagram, but first of all, let's have this introduction from this. Yeah, one more thing. The plasma membrane allows some molecules to move freely through it, and some molecules are allowed with difficulty. So molecules which are soluble in water, they have a lot of difficulty moving across the plasma membrane, while molecules which have easy solubility in lipid, fat soluble molecules, they can be very easily transported. Water soluble molecules have some difficulty because the plasma membrane is mainly made up of lipids. That is the reason. So if something is lipid soluble, it will easily cross. But for water soluble substances, we have to introduce some special help. So some molecules can move freely, water can move freely, carbon dioxide, ammonia, oxygen. Small molecules can move freely. But for larger molecules like sodium ion, potassium ion, chloride ion, we need some carrier transport molecules. And this is nothing but proteins. So proteins form a channel, and which within these things they can move freely. So this is a small diagram. It's not a very small diagram. I'll give this slide to you so you can study this. Uncharged molecules can move freely. Lipid soluble molecules can move freely. Water soluble molecules need a carrier to move, and ions also need a carrier. So it might be passive. It might happen by itself, or it might need the help of some molecules. And when we need some help from molecules, they are normally done with the help of ATP. So at the cost of ATP, spending ATP, you can do that. Okay. So this is something else. Right now, cell wall, cytoplasm, all these things. First of all, let's continue with the endoplasmic reticulum. So what is endoplasmic reticulum? It's a transport substance or a manufacturing factory also, and it consists of a interconnected membrane network of tubules. Okay. Endoplasmic reticulum is a interconnected network of tubules, and you can see in this figure here how this endoplasmic reticulum is represented. It can also be of two types. If it is having ribosomes, then it is called rough endoplasmic reticulum. If ribosomes are absent, it is called smooth endoplasmic reticulum. Smooth endoplasmic reticulum does not have any ribosomes. Rough endoplasmic reticulum has ribosomes. So rough endoplasmic reticulum makes protein, and what does smooth endoplasmic reticulum make? Smooth endoplasmic reticulum makes lipid molecules. Okay, it makes lipids or it makes carbohydrates. Okay, so first of all, let's continue with endoplasmic reticulum. Endoplasmic reticulum, please write, it is an interconnected network of tubules closely associated with nucleus. Tubules closely associated with nucleus and with Golgi apparatus. Closely associated with the nucleus and Golgi apparatus. It may be of two types. It may be of two types. Rough endoplasmic reticulum and smooth endoplasmic reticulum. So what does rough endoplasmic reticulum mean? RER, rough endoplasmic reticulum. It is having ribosomes on its surface. It's a similar to endoplasmic reticulum. Only difference is it contains ribosomes. It is having ribosomes on its outer surface. Ribosomes on its outer surface, giving it a rough appearance. Giving it a rough appearance. It is associated closely with the protein manufacturing process. It is associated with the, its main function is protein synthesis. Protein synthesis. Its main function is protein synthesis. Protein synthesis. Nothing special about it, just protein synthesis. Now smooth endoplasmic reticulum. Smooth endoplasmic reticulum. Smooth endoplasmic reticulum. It does not have any ribosomes on its outer surface. It does not have any ribosomes on the outer surface. Okay, giving it a smooth appearance. Giving it a smooth appearance. It is associated with lipid biosynthesis. Smooth appearance. Right? It is associated with lipid synthesis. It is associated with lipid synthesis. Okay. Now let's put the heading Golgi apparatus. Okay. So what is a Golgi apparatus? Golgi apparatus is the packaging process, packaging and processing center. It is involved in synthesis of all the substances that are manufactured in endoplasmic reticulum. All the manufactured substances, they will enter here. It's an interconnected network of cisternae. It's a huge network, and the network, you can see here, the tubules, they are not present. What is present is a large system of wash basin like structures, and these are called cisterns or cisternae in plural. Okay. So molecules come from endoplasmic reticulum or other organelles in vesicles. They fuse with Golgi membrane, and they might be modified or added with a carbohydrate residue or something else, and then it is modified by Golgi apparatus, and the molecules are basically budded in the separate vesicle from the last surface of Golgi body, and they may combine with plasma membrane to secrete content. So this is how this is happening. This is the total girl apparatus. This is the figure, is the total appearance of the girl apparatus. So there is a nucleus here. Nucleus is associated with the rough endoplasmic reticulum and the smooth endoplasmic reticulum, or both, or one of them. And the proteins are made by rough endoplasmic reticulum. They are forming some vesicle-like structure, bubble-like structure. It is fusing at point number 2 in the figure here. Point number 2 in the figure here, please look. It is fusing with the Golgi apparatus. It is fusing with the Golgi complex. Students, please don't talk in the middle of the class. I can see you. I hope you understand. Okay. So at point number 2, they are fusing with the Golgi complex. At point number 3, it is passing inside, and after passing inside, it enters the outside of the forming phase or the trans phase of Golgi apparatus. It is called secretory vesicle, and secretory vesicle then fuses with a plasma membrane and releases its contents outside. This is how it is happening. Okay. So please write now. Golgi apparatus or Golgi complex, it was discovered by Camillo Golgi. Camillo Golgi. It serves as the, just a second, spelling became Camillo Golgi. It serves as processing and packaging center of the cell. Serves as processing and packaging center of cell. Processing and packaging center of cell. Okay. Substances are shipped or transported from from endoplasmic reticulum to the Golgi complex. Here they are packed. Here they are packed and processed and packed, and finally they come out from the other end as secretory vesicles. Okay. Now these secretory vesicles will reach their destination. Secretory vesicles. Okay, so this is the main function of Golgi complex. I believe I cannot finish it. Let me see what I can do. Now, please write about Golgi body. Golgi complex is an interconnected network of cisternae. Endoplasmic reticulum is an interconnected network of tubules, and Golgi body is an interconnected network of cisternae. Interconnected network of cisternae. Okay. All right. So the last term we have to do today, it is a lysosome. What is a lysosome? Lysosome contains digestive enzymes. It contains all acidic hydrolase enzymes. It contains carbohydrate digesting enzyme, lipid digesting enzyme, protein digesting enzyme. So it can literally digest anything within the cell. Within the cell, it is capable of digesting each and everything. Right? So please write now. Lysosome also called suicide bags, the lysosomes are filled with acidic hydrolase enzymes. Acidic hydrolase enzymes. They can digest any substrate. They can digest any substrate. They help in recycling various organelles. Various organelles. So there are substances which make the membrane of lysosome very much unstable, and there are substances which make the membrane of lysosome stable. So if it is happening by itself, it is called programmed cell death or apoptosis. Lysosomes can burst within the cell and destroy the cell from the inside. Okay, or they can choose not to burst. So both these things are different phenomena. So I believe I can cover this in the next class. So Girl apparatus has been finished now. In the next class we will deal with vacuole, nucleus, mitochondria, chloroplast, plastids, centrosome, flagella, cilia flagella, centrosome, centrioles. All these will be covered in the next class. So in the next class, this chapter will get over. Okay. So I'll also send you the slide along with it. Okay, not right now, but I'll send it later. Okay. So thank you, dear students. Alright, so in the next class when we'll meet, we'll have some questions. We will have some queries regarding this. And in the next class, we'll finish this chapter and then we'll move on to the biomolecules and the cell division part. I believe that biomolecules has been covered by ma'am, so I'll probably take cell division. Okay. So thank you, students. Thank you for your time and good day ahead. Thank you.